Flagellin, the structural protein that makes up the filament of bacterial flagella, is emerging as a key molecular bridge between the nervous and immune systems, especially in the colon, where microbial density is highest and immune sensitivity is most acute. Long considered merely a microbial signature for immune detection, flagellin now appears to play a dual role. It not only alerts the body to microbial motion, but also engages with gut sensory circuits involved in hunger, satiety, and local inflammation. This makes it a central figure in understanding both appetite regulation and chronic gastrointestinal conditions like inflammatory bowel diseases, IBD. The body detects flagellin primarily through toll-like receptor 5, TLR5, which is expressed on intestinal epithelial cells, resident immune cells, and even on certain enteric neurons. When flagellin binds to TLR5, it activates signaling pathways that lead to two distinct but tightly interconnected outcomes: one metabolic, one immunological. On the metabolic side, flagellin's detection in the colon influences hunger regulation through its interaction with enteroendocrine cells and neuropods, the gut's sensor transmitters that can synapse directly with the vagus nerve. When stimulated by microbial flagellin, these cells can trigger the release of satiety hormones such as GLP-1, glucagon-like peptide-1, and PYY, peptide-YY. These hormones not only slow gut motility and reduce appetite, but also engage brain centers that regulate reward and impulse control. This real-time signaling loop helps the brain interpret microbial activity as a proxy for digestive status. When motile bacteria are abundant and actively metabolizing food, the gut reports fullness to the brain, reducing the drive to eat. At the same time, flagellin, TLR5 signaling activates immune pathways in the colon, especially through the NF-B cascade, which can upregulate cytokine expression and modulate local immune tone. Under healthy conditions, this signaling serves a homeostatic role. It helps the gut monitor microbial traffic, maintain barrier integrity, and fine-tune tolerance versus reactivity. The colon, constantly exposed to bacterial products, relies on this nuanced detection system to balance vigilance with restraint. But this balance is fragile. In genetically predisposed individuals or under stress, TLR5 activation by flagellin can become exaggerated, leading to immune hyperreactivity, chronic inflammation, and disruption of the mucosal lining. This is precisely where insights into inflammatory bowel disorders such as Crohn's disease and ulcerative colitis become relevant. Studies have shown that patients with IBD often exhibit altered flagellin reactivity, with elevated antiflagellin antibodies, dysregulated TLR5 expression, and a microbiota skewed toward motile, pro-inflammatory strains. These changes suggest that what is normally a regulatory signal has become a misinterpreted threat, leading to an immune overreaction that damages the gut lining and perpetuates the disease cycle. Interestingly, the same dysregulated signaling may also impact appetite and weight regulation in IBD. Many patients with active disease experience reduced hunger, early satiety, or even food aversion, which may in part stem from chronic flagellin exposure stimulating satiety pathways via TLR5. Conversely, during remission, reduced microbial motility or altered microbial composition may blunt these signals, potentially contributing to weight gain or metabolic disruption. This fluctuating microbial neuroimmune interface may help explain the cyclical appetite disturbances that are so characteristic of IBD and other gut-brain axis disorders. Moreover, experimental work in mice shows that TLR5 deficiency, which prevents normal flagellin sensing, leads to microbial imbalances, low-grade inflammation, and metabolic disturbances, including obesity and insulin resistance. These animals often have a gut microbiota that shifts toward opportunistic, flagellated bacteria, suggesting that flagellin sensing not only responds to microbial activity, but also shapes microbial ecology through feedback loops involving both immunity and neuroendocrine function. This makes TLR5 and its interaction with flagellin a strategic therapeutic target. Modulating TLR5 activity, either through selective antagonists to reduce hyperinflammation or mild agonists to promote tolerance and healthy satiety signaling, could offer novel interventions for IBD and appetite dysregulation. Likewise, strategies to reshape the microbiota, reducing pro-inflammatory motile species while supporting microbial diversity and SCFA production, may help restabilize this dual sensing system. The vagus nerve, as the primary conduit of gut-to-brain information, may also be leveraged here. Enhancing vagal tone through non-invasive stimulation techniques or behavioral interventions like deep breathing, cold exposure, or dietary modulation could help recalibrate the neuroimmune dialogue disrupted in chronic colitis or Crohn's. This suggests that multimodal treatments targeting microbes, immunity, and gut-brain signaling may be more effective than immunosuppression alone. In summary, the interaction between flagellin, TLR5, and gut neural circuits represents a convergence point for hunger control, immune balance, and disease pathogenesis. In health, this system allows the colon to fine-tune digestion, defend against infection, and regulate appetite. In disease, it becomes a distorted echo chamber where microbial motion is mistaken for danger, satiety signals become dysregulated, and chronic inflammation takes root. Understanding and restoring the balance of this neuroimmune interface is not just a scientific challenge. It may be the key to unlocking better treatments for some of the most complex and debilitating disorders in gastrointestinal and mental health.